Namaste students. First of all, I, JVWU Dr. Y. Chandrakala, Assistant Professor, Jyoti Vidyapeeth, Women's University, Jaipur. In today's session, we are going to discuss about the endocytosis. Endocytosis is a cellular process in which the substances are brought into the cell. The material to be internalized in the surrounded from the surrounding area of a cell membrane. Cell membrane is the biological membrane that separates the interior of a cell from the outside environment and which protects the cell from its environment, which then bursts off inside the cell in the form of vesicles, contains the ingested material. Endocytosis includes pinocytosis, the cell drinking, or the phagocytosis, that cell eats. Okay, it in the form of active transport. The term was proposed by D. Group in 1963. The phagocytosis was discovered by Ilya. Endocytosis pathway can be subdivided into four categories. That is the receptor-mediated endocytosis and is known as the clavellar pinocytosis and phagocytosis. So, catheter-mediated endocytosis is mediated by production of small vesicles that have a morphological characteristic coat made up of the cytosolic protein caliphrines. Caliphrine-coated vesicles are found in virtually all the cells and form a domain of plasma membrane, termed caliphrine-coated pits. Coated pits can concentrate large extracellular molecules that have different receptors responsible for receptor-mediated endocytosis of the ligands. Example, low-density lipoprotein, transferrin, growth factors, antibodies, and other. Cleverley is the most common reported non-calcium coated protein membrane, which exists on the surface of many, but not all the cell types. Okay? So here this they consist of the cholesterol binding protein clevelon VIP 21, which with a bilateral enriched cholesterol and a glycolipid. Okay, so cleverly are small, approx 50 nanometer in diameter, and the flask shape in the membrane that resembles the shape of the clay. They can constitute up to a third of the plasma membrane area of a cell. Some tissues being specially abundant in smooth muscles. So here the type 1 pneumocystis, fibrocytes, or adipocytes, or endothelial cells. Uptake of the extracellular molecules is also believed to be specially mediated via receptor. Photocytosis is the form of the receptor-mediated endocytosis that use the Kevlar vesicles to bring the molecules of the So the photocytosis is the form of receptor-mediated endocytosis that use the clevellar vesicles to bring the molecules into various sites into the cell Unlike most endocytosis that use the clavella derive the content of the vesicles. Okay. So the pinocytosis, which usually occur from the highly rough region of the plasma membrane, is the invagination of the cell membrane to the form of pocket. Phagocytosis is the process by which the cell binds to the internalized particular matter large than the around the 0.7 micrometer in diameter. Okay. So the most recent experimental here the suggests like the cathepsin-dependent endocytosis field has been lacking. In the study shows it regulates by highly prevalent cathepsin-independent endocytosis pathway known as the CLIC and GEEC pathway. The principal components of the endocytosis pathway is the mammalian cell consisting of the distinct mammalian compartments. Okay. So early endosome, late endosome, and lysosome. So the cathepsin-mediated endocytosis have a major role of endocytosis in the plasma membrane. So large protein assists the protein code with. Okay. So the fourth pith of the vesicles was first seen in a thin section in an electron microscope by Arthur George. This session is powered by 2.0 Jyoti Vidyapeeth Women's University, Jaipur. If you have any queries, we'll discuss in the problem solving session.